DNA replication. After Watson and Crick published their paper on DNA structure, they also published a paper on their hypothesis of how DNA would replicate. And they said that the hydrogen bonds would break in between each strand here, and each strand would serve as a template for a new strand. And this is called semi-conservative replication. And you can see the original strands yellow, new strands red, new strands green, new strands black. There were other models out there, and Nielsen and Stahl designed experiments to distinguish between these models, and their findings supported the semi-conservative model. Replication takes less than an hour in E. coli and a few hours in humans, and you're copying a lot of nucleotides during that time. But even though you're copying that many, only about 1 in a billion nucleotides will have an error. And in the next video, we'll talk about how those get fixed. So, you have an origin of replication here. And it starts out as just a little bubble. And what happens is a protein comes in and separates the two strands to make it a replication bubble. In eukaryotes, there are multiple replication bubbles. And so replication is taking place at multiple places down the chromosome. And as the strands are copied and extended more and more, they eventually fuse, and at the end of the replication bubble, you have a replication fork, which is the region where they are being separated. And you have enzymes that come in and separate these two strands. The main one that's going to separate them is called helicase. It unzips your genes. And then you have a single binding protein that comes in to keep them from sticking back together. And because they're uncoiling at the bubble, it makes the coiling on either side of the replication forks much tighter. So you need topoisomerase to come in and relieve that strain from twisting. And topoisomerase will actually break the DNA, turn it, and stick it back together. Once untwisted, the enzyme that comes in to make new DNA can actually start. But first you're going to need primase. Primase is your starter enzyme, and it makes a short chain of RNA from a single nucleotide. And once the primer is 5 to 10 nucleotides long, the new DNA will start at the 3' end of the DNA primase. So here you can see the 3' versus 5' ends. This is based on the carbon structure, the carbons within the sugars. And DNA is said to be anti-parallel, that you run 3'5 to 5' this way, 3'5 to 5' this way. And it's just those carbon or those sugar atoms are in the opposite direction. Here's a close-up of which carbons would be. Here's 1, 2, 3, 4, 5'. Lots of chemistry, fun stuff. Now, once you start making the strands, once you have that primer, DNA polymerase 3 can add nucleotides to the 3' end of the primer on the leading strand. And the leading strand, you can see here, is nice. It's continuously just adding nucleotides, but the lagging strand is discontinuous because it's having to work in the opposite direction towards this replication fork. And as the replication fork opens more primase is needed to start the strand. Once the strand is complete, another DNA polymerase 1, comes in and replaces the DNA, and then ligase goes in and joins the sugar phosphate backbone to make it a continuous strand. The end.